Last day, Wednesday, I started with composition of components of blood, composition of blood, and their functions. And that class I couldn't finish, particularly on the cells. So after completion of this, I will try to cover those parts. Okay? Otherwise, you will not be able to answer. Then I will start EC. Instead of 3 hours, it may be of 2 hours. Instead of so many questions, I will decide how many questions from blood and its component from plasma can be placed to you. What is the competency? In your topic hematology, the second competency is plasma proteins. What you have to know about their origin, their forms, types, okay, and you know them. Most of you are acquainted with these classifications. Variations. Before that, we have to know those different forms are having normal values, a range they are having. What happens if these values go beyond range, either below or above? What are these conditions called? And in which pathological conditions these are seen? Important. That means variations. The variations of plasma, they are in their contents. What are these variations and what are the conditions? What are the pathological conditions, clinical implications? You have to know. That is the vital part of it. So you have to concentrate on that. First, you will be knowing what is there. Every component has a specific functions. And if you know the functions, then if its level is increased, its level is decreased, then you can assess, then you can predict what may be the consequences. So that's why you have to, I think it will be better if you come forward. All this work. So the learning points, here are the points I will tell. There are other points too. These are not exhaustive. Other than this, I will also discuss some other points. Okay? These are the competencies. I have shown it, second, which is been shown there, competency number 2. Now from this, you can see what is this? Blood is taken, anticoagulant is mixed, we have centrifuged it, now plasma and cells were separated, okay. So different layers, the topmost layer is plasma, and after that there is a thin coat, buffy coat. What you will get in this coat? Platelets, WBCs, and abnormal cells, cancerous cells, the cells which were not supposed to come from the bone marrow, they are in the primitive stage. Myeloblasts, blast cells, they are not supposed to come to the circulation. If they come under pathological conditions, say in cancerous condition, so you may find those cells in this buffy coat. Okay? So this layer is very important for pathological purposes. So at the bottom is cellular cells, and these cells are constituted, this layer is constituted by only RBCs. Okay? We will discuss the RBCs we classify them in 2 ways. According to their size and according, the RBC contains one pigment, red pigment. What is the name of this pigment? Hemoglobin. So hemoglobin is a right color pigment. What is the chemical nature of it? Anybody? Okay. Let's stop it here. I want to ask you one question. What are proteins? What are proteins? Proteins are organic, nitrogenous, macromolecule, colloid in nature, made up of nitrogenous compound. The nitrogen should be there. If there is no nitrogen, it is not protein. So it is a nitrogenous compound, macromolecular, colloid in nature, an organic compound. Okay, it cannot pass through cell membrane normally because through the semi-permeable membrane they cannot pass through. So these proteins are made up of numbers of different types of amino acids. All amino acid different kinds, and all of you together formed a protein. By joining your right hand and left hand, right hand is this way linked to all of you. And this link is called peptide bond. All the amino acids must have two things. One is amino group, another is carboxylic group. Right hand, amino group, left hand. So amino group, carboxylic group, they can join together, form a peptide link. In this way, they are linked. Now the proteins are having different type of structures, primary structures. Only if the amino acids are linked together, only by peptide bonds, they are called simple primary structure. But among you, some amino acids have affinity for another amino acid, and she is sitting there. So you will be trying to come closer. Okay? So this is the chain. Okay, going. And you saw your friend is here, you are here. So both of you will be coming together, forming a bond. This will give you secondary structures. In this way, I will not taking the chemistry of protein. So tertiary structures, quaternary structures, very important. If their quaternary structure is disturbed, their activity, their function will be disturbed. They will be of different kind of protein. A particular protein, say globin. They are in the globin of hemoglobin, globin, these are protein. There are 4 chains, 2 alpha chains, 2 beta chains. All the alpha chains are having 141 amino acids. Beta chains are having 146 amino acids. Not only this. First amino acid is this, second must be this, third must be this, sequence must be maintained. Who will be after whom? If the sequence is disturbed, replaced by, then it is not a normal globin. We will find there are different types of abnormal hemoglobin because of this. Somebody intentionally replaced you by another amino acid, but it has changed its nature and it is a pathological abnormal globin, abnormal hemoglobin. Okay? I am deviating to another direction, coming back. Hemoglobin also a protein. Proteins are of different kinds. They may be classified simple protein. What? Simple protein means if you hydrolyze this protein, you will get only amino acids, nothing else. So these are simple proteins. And some are conjugated protein. Simple protein is associated with, linked to another non-protein substance, then we call it complex protein or conjugated protein. Okay? That is protein, it is linked to another non-protein substance. It may be metalloprotein, it may be glycoprotein, it may be lipoprotein, means lipid and protein, carbohydrate and protein, glycoprotein, metal plus protein. Lot of enzymes are there in our body. Without this metal, they are not able to function. Okay? So, these are metalloprotein. And our hemoglobin, another is derived protein, another class is derived protein. In nature you won't get them. But when you intake a protein, this will be degraded, no? So, one protein from there, another compound will be formed, from that another smaller compound. So, these are the derived product. Acid protein, acid metaprotein, these are derived products. Peptides, these are derived products. Okay, these are not present in the nature. These are produced by the body from their degradation in the digestive system. Okay, these are the different class. Now our hemoglobin, is it a simple protein? Is it a conjugated protein or it a derived protein? Simple. Hemoglobin. What is that? Globin is the protein. As I said, there are 4 chains. 2 alpha, 2 beta. Okay? And another part, non-protein part, heme. And heme is made up of 4 pyrrole rings. These pyrrole rings are attached to iron, forming heme. Now the iron is linked to one of the peptide bond, one of the chain. Now they are hemoglobin. So hemoglobin is a what type of protein? Conjugated protein. More precisely, it can be called metalloprotein, or we can say it is a chromoprotein. Why chromo? A red color pigment is attached to heme, is the red color pigment. It is attached to globin. So when I will be asking, what is hemoglobin? It is a protein. What type of metalloprotein? What type of protein? It's a conjugated protein. What type of? Either you can say it is a metalloprotein, or you can say it is a chromoprotein. Clear? In one molecule of hemoglobin, if you analyze one molecule, remember all these points particularly. One molecule of hemoglobin is having 4 hemes. That's why we call it HB4. And all the heme is attached to one chain. So all the 4 will be having 4 chains, and out of these 2 are alpha chains and 2 are beta chains. If the heme, it is the heme portion, it is the iron that binds with oxygen to form oxyhemoglobin. Now tell me, how many oxygen molecule can be carried by one hemoglobin molecule? Yes, correct. Since there are 4 hemes, everybody can attach to one oxygen molecule, there are 4. So HB4OH. Oxyhemoglobin formula HB4OH. Understood? Okay. When all the 4 hemes are saturated with, all the four are having oxygen, then we call hemoglobin is 100% saturated with oxygen. Okay? So remember this point. So coming to this. Here cells are more. It happens it is called polycythemia. If it is more than normal, it is called polycythemia. And if it is below normal, erythrocytopenia. When it is seen, when RBC is less, it is seen in anemia. So when you find that this layer is less, PCB is less, hematocrit is less, what does it indicate? There is anemia. Do you think their percent is less? Do you think their number is also less, or number is same, or number is more? Number is also less. That means PCB is less. When PCB is there, their count is also less. Now this service we can classify anemia in different forms. One is etiologically. What type of classification? We'll take a class on this. Okay. We can classify anemia etiologically. How it developed, why it developed, what is the reason behind. So that we will try etiologically we can classify. And another type of classification is there. We classify the RBCs by morphological change. So one RBC, it is having a definite shape. We know the size, we know the shape, and we know this cell contains the hemoglobin. Every cell must have a definite amount of hemoglobin. Size is normal, but hemoglobin is less. Normocytic, hypochromic. Now the cell, the RBC will be regarded as normocyte, normal in size, but their hemoglobin content is less, that means hypochromic. Or if the content of hemoglobin is normal, then it will call normocytic, normochromic. If the size is less than normal, then it is microcytic. And normally it should contain hemoglobin. If the content of hemoglobin is normal, then microcytic, normochromic. And if the hemoglobin is also less, normocytic, hypochromic. Again, another type of RBC you will get macrocytic, but normochromic. In a normal size RBC, the amount of hemoglobin there should it have the same amount is given, but the cell is bigger. So this is what? Macrocytic, normochromic. So we will classify them when we will study anemia, different types of. Say, if you don't take vitamin B12, if you don't take with your food vitamin, folic acid, if you don't take iron containing food, what will be there? Less RBC production, less hemoglobin production. So there will be anemia. These are called nutritional anemia. It is related to another condition may develop. Your blood cells are gradually breaking down. Hemolytic anemia. So different kind of anemia we will study. So it is a very important question, anemia. What type of question? Short note, 5 marks. So you have to study in physiology, in pathology, everywhere you have to study it. So study it from now only. 90 to 92% is water. Average 91%. Average 91%. I will ask, if the percentage amount of water is less than normal, less than 90%, what will happen to your body? What is the condition called? I will ask. Not now. Then solids. So remaining 9%, average 9%, remaining 9% is solids. So out of these solids, there is another component, gases. Blood contains different gases. No? We assess PO2, PaO2, partial pressure of oxygen in artery. So PbCO2, partial pressure of oxygen in venous blood. So we know blood contains gas. So it is also a component of plasma. So it should be there. Oxygen. The main function of one of the main function is of the blood is to supply oxygen to the cells. Otherwise, cell will die within few seconds, within a few hours. So how it will go? Through plasma. Okay. Either as physical solution, very less, and mostly as chemical compound, oxyhemoglobin. So they should be there. Oxygen. Carbon dioxide should be there. Nitrogen should also be there. So these are the gases present in plasma. Now let us come to solids. And the solids, you will get two things, organic and inorganic. Very good. So again we will classify this into inorganic and organic. Okay? Out of this, 1% is inorganic and 8% is organic. Who can tell? The inorganic constituents. Without these inorganic constituents, you won't survive. There are so many inorganic constituents. Everything is important. What are these? We can classify them, these electrolytes, as cations and anions. Good, very good. Study well. So these inorganics are two types. One is anions and another is cations. Example of cations. Okay. Sodium. Is it needed for us? What for? If the concentration of sodium in the plasma is less or more, it is a pathological condition. Serious consequence. You may have to be admitted in ICU. Potassium, if it is changed, your heart may not work properly. So it is a serious condition. So why? They are very important in maintaining the action potential in performing. And if action potential is not developed in my body, your sense, how you will sense everything? Nothing you can sense. Nothing you can do, nothing you can sense. You cannot maintain the resting membrane potential that exists in the cell. Across the cell membrane, there is a potential difference, which is called resting membrane potential. And this value is definitely known. It is constant. When it is disturbed, action potential is developed. And we disturb it by stimulus. Who can tell what is stimulus? You might have heard, everybody might have heard. Stimulus, what is stimulus? Very good, excellent. Any change in your environment, be it external environment, be it in your internal stomach ache, what happens? Why stomach ache? When you take too much of a spicy food, when acidity develops, you feel pain, your menstrual pain. So these are change in the internal environment. External environment, I am talking to you. This is a sound. This sound is hammering your ear. Okay? And you are sensing what I am telling. And you are seeing me, your eyes. Okay? So, when I came, environment changed. When I will be going out, environment changed. Accordingly, you will respond. So, these are stimulus. It may be electrical. It may be magnetic. It may be light. It may be sound. So, stimulus can be of different types. Okay? With the change, with the stimuli, we respond. We respond. You might have one response. What is? Fight or flight. When? Suppose you are going somewhere, suddenly one snake. What is this? Most poisonous? For example, cobra. What you will do? You will fight with this? You will run away. Okay? So you will not fight. Somebody tried to hurt you. So first you will run away. Then you came to a room and he also entered and there is no escape route. So you are there. What will you do? For your survival, you will fight. So, our response, either fight or flight, depending on what our nervous system decides. If our opponent is very strong, we will fly. If it is weak, okay. So, I wanted to tell that all of these are very important. Be it is sodium ion, potassium ion, calcium ion. Without calcium ion, blood will not be coagulated. Without calcium ion, your muscle will not be contracted. So, without calcium ion, I can think. It has got some enzymes require calcium ion for their activity. So, calcium ions are very important. When calcium ion concentration, hypercalcemia, hypocalcemia, what can be the consequences? These are the clinical implications. You have to know. Okay? You have to know in pharmacology. You have to know in pathology. You have to know in medicine. Magnesium. Is it needed? These are also needed. From ATPase enzyme, the activity of the ATPase. ATP, our currency. If your currency is from Bangladesh, you can't buy anything. If it is Indian currency, you can buy anything. So with this you can buy many things. So this is the currency which is needed by all cells for their activities. And first what will be, how it will be doing its activities? First ATP will be broken down. Every ATP has 3-phosphate in it. That's why adenosine triphosphate. These are high energy phosphate bond. High energy phosphate bond. When this is broken, what is formed? ADP plus 1 inorganic plus some energy. With this energy, I will act. This energy will be utilized for the function. Sometimes I need this breakdown here. So what will the AMP will be formed and pyrophosphate will be formed. We'll see in biochemical pathway in many reactions, ATP is broken down, AMP is formed, pyrophosphate is formed. You will find in metabolic pathway. So that's why magnesium is important. Copper. If excess copper is deposited, can be deposited by ceruplasmin, one type of plasma protein, carrier of copper. What is the name? I told that carries copper. Ceruplasmin that carries copper. And if this copper is deposited in liver or brain, serious condition, the patient may die. Okay, so, but copper is also very important for body functions. So these are cations. Zinc is also there for immunity. Now the anions. Bicarbonate ion, chloride ion, phosphate ion, sulfate ion, protein. Protein is organic compound, but it can be ionized. So don't take it here. So protein will be in organic constituents. Every anions are very important. Any variation, pathological condition. It will create havoc in different organs, particularly brain and heart, in other tissues. So all these are important. You have to know normal range of these ions, be it anion, be it cations. Because small variation beyond range, it is going to affect your body. Okay, now I am coming into organic. One component, others. So who will be here? Who will be here? Protein. Most vital organic component. Major organic component. Out of 9% of solids, 7 to 8% is protein. And these are performing very important functions. I will be describing one by one. You write it in your body, or sometimes only one function. You will be asked to narrate only one function. So every function you have to learn carefully. Okay, so proteins. How much protein we are coming into our lecture? How much protein we are having? 7% what? 7% of total plasma. And this one, if you take 100 ml of plasma, in this 100 ml, you will get 6 to 8 gram. Remember, protein is always in gram. Sometimes what happens? Tell me in biopsy, tell me the normal content of plasma protein. And the answer is 6 to 8 milligram %. Is it correct? No, because these values are in gram. Remember this point, okay? Always. You. Tell me, what is the normal heart rate? Get up. When I will be asking you a question, you should. Yes, black shirt. Huh? What is the normal heart rate? Normal heart rate. Don't look at normal heart rate. Huh? 7 to 80. Okay. What is the normal heart rate? Huh? 70? This is the answer. As I said, milligram, gram, in beat there is 1 unit. Without unit, the value is less important. So you have to tell 70 to 80 beats per minute. Yes, always. That's why I asked this question. Whenever if there is unit, tell the unit. Otherwise you will get 0. Without unit, no value. Same mistake you will do it, I tell you. Same mistake you will do it. In another class, if I ask this, same question will come, same answer will come, 70 to 80. Or 72. You will not be uttering or telling the unit, but you have to tell this. What is the normal respiratory rate? You, second man. Huh? Normal respiratory rate. Good. Okay, you sit and count how many times you breathe in 1 minute. You sit. Hello, with mask. Yeah. What is the normal respiratory rate? You haven't studied? I can't hear you. What is the link with this? You are sick, that I understand. But this knowledge, what is the normal respiratory? You brought it from class 10, class 11, 12, isn't it? You tell.
It is 12 to 18 breaths per minute.
Like you, he was not studying this respiration, but he had previous knowledge. Class 12 knowledge, class 11 knowledge. So, you have to tell unit also. Breaths per minute. Very good. So, I wanted to give you emphasis whenever you tell the values, always tell the unit together. So, proteins, these proteins are of 3 types. In some books, you will find 4 types. What are these? Albumin, globulin, fibrinogen. Okay? What is if you take 100 ml of plasma, how much albumin you will get? 4 to 5.5 gram %. Their amount is highest. And whose amount is lowest? Fibrinogen. Remember this. And next to albumin is globulin. When you are infected, this amount will be increased. When you are normal, it will be within normal range. When you are infected, you have to fight against that. So this content, its amount will rise. Then we will use a term. When normal amount of globulin in plasma is raised, what will be the term you will be using? Let us be acquainted with this. Suppose albumin, it came below 4, suppose it is 3 gram %. What you will tell this condition? Hypoalbuminemia. Hypoalbuminemia. Or if it is more than 5.5, because we will be asking questions. In hypoalbuminemia, what happens? So you have to explain it. So first you have to know what is hypoalbuminemia, what is hyperalbuminemia. So you have to be acquainted with these terms. Similarly, now you can tell if the globulin level becomes more than 3 gram %, then what is this condition? Hyperglobulin. Very good. Okay. So in this way, these are the different plasma. Another is prothrombin. It is needed in blood coagulation. Prothrombin, also a plasma protein made from liver. It is also a plasma protein and it is a proenzyme. When this will be activated, they will be thrombin and they will break fibrinogen into fibrin. Okay, to complete the process of coagulation. Okay. Among the others, you have to remember this range 0 to 3 gram %, 1.5 to 3 gram %, 4.5 to 5 gram %. Okay, in biopsy you will be asked what is the normal range of these values. Okay, next, what are the other compounds? Before coming to college, did you eat something? What you ate? I'm coming to you. What you ate? Roti, she ate roti. Now this roti has entered in your blood. It has entered in your blood. So what entered? Roti is made up of carbohydrates, starch. Carbohydrates too are of different kinds. One is polysaccharide. Okay? So starch is a polysaccharide. Glycogen that we store in our muscle and in liver, this is also polysaccharides. We have oligosaccharides. We have simple sugars. Means glucose, any other simple sugar, monosaccharide, any other. Sucrose disaccharide. Fructose, yes. Another, in milk you will find lactose disaccharide. Galactose. So when she ate roti, this is a complex thing. So we have a system, it's only one function, main function, convert this complex into simple one. So this complex roti means starch, it has been made simple, means starch is made up of glucose, lot of glucose molecules she developed in her intestinal lumen by different enzymes. Now this glucose can enter into blood, into plasma, they will be carried through plasma. So it should be present in plasma. You ate protein. Protein is made up of, it is hydrolyzed and some amino acids are produced. So they must be present. If you ate fat, so it is digested into fatty acid glycerol. It has cholesterol. So this will be transmitted, this will be transported through plasma. So this should be there. In blood there is triglyceride. How we tell he is having lot of fat in his blood? Means, what is this condition called? More lipid in blood. Dyslipidemia. Hyperlipidemia. And it is an alarming condition because this may develop into atherosclerosis in your blood vessel. And atherosclerosis means blood flow will be disturbed. So there may be clot formation, there may be blockage. So if it happens in heart, myocardial infarction. If it happens in brain, what will happen? There will be severe cerebrovascular accident. So these are very important. So in plasma, you will also get these things. Cholesterol you will get. So in other compound, what is there? Like glucose, amino acid, triglyceride, cholesterol, these you will get. Fatty acids, these you will get. And one more thing you will get, you have taken all these important things from blood and you will be catabolizing it and you will produce some unwanted things. These are excretory products. You don't want them. Go away. You will send them, the blood will send them to your excretory organs, to your kidneys. Okay? From there they will be eliminated. So they must be present in your blood. So blood should, in other compound they should be there. So what are these? Urea. When there is a problem, we are doubting there may be kidney may be affected. So immediately you will be writing kidney function test, KFT. Means what will be done? Whether kidney is performing this function. If the amount of this excretory products is more, it is not doing well. Creatine, urea. So when their level is high, that means kidney is not working. When it is amount creatine is going beyond 2.56, then you may require service of artificial kidney. That is diuresis. Okay. So that's why it is very important to study their contents of this. You understood this? Okay. So that's all about the composition of plasma. I think you will be able to remember this. Now we are coming into, so we came to know the normal different types of plasma proteins, their normal levels. Okay. Let us study one by one. There, bolo, which one you want? What to discuss? The function. So first, we learn as a whole function of plasma protein. All plasma protein together, they will be performing some functions. Okay, near about 9 functions they perform. And after that, we will be learning separately. You are albumin. What is your function there in plasma protein? You are globulin. Tell me. What? Don't bring her. You must be having your specific function. Tell me, globulin, your functions. Tell me, fibrinogen, your individual function. Tell. Understood? So we have to now learn as a whole what are the functions of plasma proteins and at the same time individual function of individual plasma proteins. Okay? What I checked? Whether pallor is there. Pallor means the capillaries whether are having adequate hemoglobin or it is pale. Less hemoglobin, it will look pale. When it is pale, he or the person may be suffering from anemia. Isn't it? Okay. I thought he is having anemia, actually not. Okay. So now let us come to general function of plasma protein. Okay. What are the general function of plasma proteins? First one, maintenance of osmotic pressure. Sir, we have doubt. What is osmotic pressure? Do you have doubt? No doubt. Osmotic pressure? Who can explain in short what is osmotic pressure? Okay. Before that, actually, if you don't have, why I am asking? Sir, it is class 12, 11 in chemistry, physiology, you have learned it. But if you don't have clear conception, then how can you understand this function? What is osmosis? Forget osmotic pressure. What is osmosis? Ajo. Today I am having lot of time. So that's why I am going slow. Osmosis. Raise your hand. Who can explain osmosis? Oh, you. Yeah? You. Okay, very good. Come. Help me. Good, very good. First you clear.
To lower concentration across a semi-permeable membrane.
Achha.
So my
You explain.
handwriting is very bad.
Huh?
My handwriting is very bad.
Okay, we tell him he will write. What is solution? Sir, what sir is asking us? What is solution? Is it a question? But we are not able to answer. What is the solution? Solution. Not mixture. Mixture is different. Solution. What is solvent? In a solution, there is solvent, there is solute. Solvent is the medium in which the solute is dissolved. Okay? So now it is a solution. Okay? The solute will not be sedimented. They will be remaining within this. So this is a solution. See, now a solution can be hypotonic, can be hypertonic. Isn't it? If the concentration of the solute is more in comparison to another solution, if the ওরে কি লিখছিস? Okay, okay. আমি বলেছি explain করতে. I wanted to explain.
I can.
You can. Very good. Explain करो.
Can I draw also?
हाँ, sure.
So here we have
Two solutions.
yeah, two solutions.
Solution A, solution B.
Yes, sir.
Which one A, which one B?
So this is
A, this one B. So these are solvent molecules
So the smaller ones are solvent molecules.
and the bigger one
are
is
solute.
solute. Here we can see, here in B we can see solute is less. Solute is less. Yes, solute is more. So which one is hypertonic? This is hypertonic in relation to this, having higher concentration. This one is having
Lower concentration.
lower concentration, means hypotonic. And this is a membrane, semi-permeable membrane. What does it mean?
So semi-permeable membrane allows only the solvent molecules to pass through it. And
Ah,
doesn't allow the
only no solute molecule can pass through, only solvent. If it is water, only water can pass through. Now, if you keep it for some time, what you will find?
So the
Both became isotonic. The number of solute molecules here per unit area, here also per unit volume is same. These are now isotonic, same concentration. How it happened?
so through osmosis through
From here, some solvent molecule entered here. Water entered here. And that part was more concentrated. Now, because of entry of solvent, it is gradually becoming diluted, and that one becoming concentrated at one time. At a part where they will be, their concentration will remain same. Why? Because of entry of water into this, into the concentrated one, hypertonic solution. That means the hypertonic solution applying a force on the hypotonic solution, hey, give me water. That means hypertonic solution, whenever it comes, this solution is hypertonic in relation to this. That means this hypertonic solution requires water. It will collect water from this water drawing pressure. So always remember, this is nothing but water drawing pressure. It is thirsty. It wants more water, more concentrated, more water. Got the idea? Now you can see hypertonic solution is drawing some solvent with a pressure. It is drawing this fluid with a pressure, and this is osmotic pressure. And how much is this pressure? Again, you create pressure here. The water will be going back. How much you have drawn? This much. So you send it back. So you will require a pressure. That is the osmotic pressure. Yeah. Good. I think now you will not forget what is osmotic pressure. Okay? So, now our plasma protein, their function is to create osmotic pressure. And the osmotic pressure created by plasma protein in the blood, in the plasma, is also known as oncotic pressure. What? Osmotic pressure. They are creating a pressure. Can you tell me what is this pressure called? This osmotic pressure created by the plasma protein, also called understood? Why are you? I brought you from there to here. You were not listening. And the normal osmotic pressure is, oncotic pressure is, what is the unit of pressure? Millimeter of Hg, mercury. 25 millimeter of mercury. How much? 25 millimeter of mercury. Okay? What happened? Swelling because of accumulation of fluid. Where? In extracellular, outside the cell, in between the cells, in ECF, extracellular fluid. Why? Because of that very osmotic pressure. The osmotic pressure is not normal. It is abnormal. Why? Plasma protein is abnormal. The plasma protein content is abnormal. That's why this is developed. Whenever you find a swelling, overall swelling in different parts, that is, this is called edema. And why this edema? There may be in various conditions. One of the reasons is alteration in plasma. But if plasma protein level has came down below normal. As a result, osmotic pressure is reduced. Now what happens? How it develops? Let's see. I'll ask this. I'll ask you try to understand this point. Every cells, we know, in the first class, we told you, blood is the life of the cell. Without blood, cell cannot survive. Because all the required things for the cell, for the tissue, will be supplied by blood. Okay? So suppose this is one cell of my body. So it has to be supplied with blood, isn't it? Otherwise it will not survive. It should get all important things like glucose, amino acids, fatty acid, hormones. Oh, in constituents, I forgot. Hormones are also there in constituent, in other compounds. Hormones, enzymes, these are also there. Okay, vitamins, these are also there, I forgot. These are also there in other compounds. So, everything has to be supplied. So, that's why blood is coming. And this is the capillary. This is the capillary. So, how capillary is formed? Artery, arteriole, capillaries. So, this is the capillary. This is the arterial end of the capillary. And from this capillary, this should have been in red color. Venous end of the capillary. In the arterial end, blood is flowing, and this blood must have a pressure. Otherwise, how it will flow? If somebody doesn't push you from the back, will you go forward? You are inert. Somebody has to pull. Somebody has to pull. Driving pressure should be there. So here also this driving pressure is created by our heart, by pumping blood. So because of driving pressure, blood is passing through. When this blood will be coming here, this blood must have a pressure, and this pressure will call hydrostatic pressure. A tube, a pipe through which water is passing. Since it is passing, so it must have a pressure. Okay. If you make a perforation on the pipe, what will happen? Water will come out because of this pressure, because of this hydrostatic pressure. Similarly, because of the hydrostatic pressure, what is the hydrostatic pressure here? 37 millimeter of Hg. So because of hydrostatic pressure, the blood will try to come out of the blood vessel. How they will come? How they will come? The wall of this capillary is made up of single layer epithelium. Single layer, one cell. Okay? Single, these are called endothelial cells. And these endothelial cells are not adhering to one another tightly like you. There is a gap in between. And this gap is called, like that gap, in between both of you. So, these gaps are called fenestra, in between the endothelial lining. So, because of this pressure, blood will try to flow through this fenestra. And what is there outside? Through this fenestra, blood will try to flow, come out. But whole thing cannot come out. The cells are bigger than the size of these pores, this fenestra. So they will not be allowed to pass. You remain in. You have other functions to perform. So be in the vessel. Plasma protein also try to pass. Oh, your size is bigger, like hathi, elephant. So you are not allowed. Glucose, okay, you go. Amino acids, okay, you go. So they will come. Their size is less. So important things will come out. These are necessary for the cells. Okay? So all important things will come out because of this hydrostatic pressure. But within the blood vessel, there is another pressure who will tell, no, I will not allow water to go out. Plasma protein. It is thirsty. It needs water. It will not allow water to go through. But who is more powerful? Hydrostatic pressure is powerful. I will not listen to you. So there will be, ultimately who will win? Hydrostatic pressure will win. And there will be a pressure difference. How much? 37 minus 25. 12. So there will be a pressure 12 millimeter of mercury with which fluid will try to come out, enter into the extracellular fluid. This is called filtration pressure. This is called filtration pressure. And these forces, hydrostatic pressure, one force. Osmotic pressure, this is also another force. So these are Starling forces. Write it down. Starling forces. Starling. Starling, S-T-A-R-L-I-N-G. Starling forces. What are Starling forces? How it allows fluid exchange across capillary. Across capillary. Now see, here two more pressure will work. Other than osmotic pressure, other than hydrostatic pressure, there are two other forces. What are these? Some fluid is already here, isn't it? They will not allow, they will allow osmotic pressure, oppose osmotic pressure. We are already a lot of fluid, we are here, so we will not allow you to enter. So they will oppose. But fortunately, this pressure, hydrostatic pressure, intracellular hydrostatic pressure, it is very less, 2 millimeter of mercury. Okay, so what was the difference? 12. And since it is opposing, so minus, it will be minus, how much it is coming now? 10. So this 10 is the effective filtration pressure. And these are the three forces. Another pressure is there. Within the cell, within the ECF, there are some proteins. They may also create osmotic pressure. And they will be favoring, okay, come. So they will add 1 millimeter of mercury pressure, 1. So the amount is negligible. So all this together gives a net pressure. If it is positive, then there will be filtration. So this is the scenario of filtration in the arterial end of the capillary. Understood? Clear? Now, try to understand every point. I will explain why edema develops. Okay? Now this blood will be entering here with plasma proteins. No loss of plasma protein. So here pressure is, oncotic pressure is 25 millimeter of Hg. But here the blood vessel diameter is relatively wider. So here pressure will be reduced. Moreover, some fluid has gone. So pressure will be reduced. It is no more 37. It is no more 37. How much it is? Here, what was the effective filtration pressure? 10. Huh? 10. Effective filtration pressure was plus 10 millimeter of Hg. And hydrostatic pressure is 17 mm of Hg. And this is the positive pressure, this is the negative pressure. Means it wants to draw and it wants to send. What will happen here? Whether fluid will enter or fluid will go out. Drawing. Osmotic pressure is the drawing pressure. And hydrostatic pressure is pushing pressure. What will be happening here? Drawing. So, what is the net pressure? Net drawing pressure. So, drawing pressure is 8 millimeter minus 8 millimeter of Hg, whereas filtration pressure is here. So, more fluid will be coming out, relatively less fluid will be entering it. Because there is a pressure difference of 2. Some fluid will be there. Every moment some fluid will be surplus there. So what will happen to this fluid? You might have heard the name of lymphatic channel, lymphatic channels. So, in the tissue space, there are lymphatic channels. Now, this extra fluid will be going through the lymphatic glands. Again, they will be coming back into the circulation. I am aging, my hearing ability is reducing. It will do effective, not filtration, absorption pressure. This will absorb. Filtration, absorption. Now there is a pressure difference of 2. As a result, some fluid will be remain extra. Okay, if would have been 10, the same amount of fluid would have entered. This fluid is having excretory products of the cell. That has by this time diffused out. Taking all these things, necessary things, carbon dioxide and other things is entering. Other excretory products entering. And some extra fluid will be retaining there. They will be passing through the now. But this lymphatics has a capacity. Beyond that, beyond a certain value, it cannot drain all the extra fluid. What will happen then? Now have a look on this. My liver is not working. I am a cirrhotic patient. I am having chronic liver disease. These plasma proteins are synthesized in liver, except gamma globulins, albumin, fibrinogen, prothrombin, and some variety of globulin they are synthesized in the liver. Since my liver is not working properly, what will happen? There will be less plasma protein. There will be less osmotic pressure. So, under this condition, my osmotic pressure came down to 25, 20. Instead of 25, it came down to 25 millimeter of Hg. Now, what is the difference? 17. Huh? More filtration will be there. Lot of fluid will be coming. And here what will happen? It is 20. Only 6. So only little fluid will be absorbed. So there will be, as a result, what will be? Lot of fluid accumulation will be there in the tissue space, resulting in this. And this is edema. I think I could explain how edema develops. Rick, is that okay? Any doubt?